Hi, everybody. I thought I would jump on here while I'm waiting for test results and kind of talk you through mitosis and kind of show you a little bit about how things are actually working. So I hope everybody's doing okay and is starting to actually enjoy their weekend. So let's go ahead and actually get started here with mitosis. Okay, so if we actually look at this, um, we kind of have an animal cell that's shown here in the first one. And so you can kind of see, like, here's the nucleus. If we kind of do a zoom in, actually, there we go. I guess it is zoomed in. Okay, you can see here's the nucleus. We've got some endoplasmic reticulums that are around our nucleus that we worked on with protein synthesis. These are probably other organelles, maybe some vesicles. And then, of course, we have the mitochondria, powerhouse of the cell. Super exciting. And so here are, you can see the chromosomes that are actually starting to, and actually pretty much have concluded that kind of supercoiling process that we actually talked about, where they would be wrapped around the histones. Here's a chromatid, here's another chromatid. So sister chromatids or brocros held together by a centromere. And they're making some chromosomes hanging out in the nucleus. You can see that the nuclear membrane is actually starting to be able to disappear here. Okay. And then the centrioles are here, and the mitotic spindle is actually, or the spindle fibers are starting to form. Um, so then, if we kind of move on to this next picture here and look at it, it kind of zooms us in even closer. We realize that part of the nuclear membrane has actually disappeared, and some of it is still starting to actually disappear. And then these centrioles, these little protein discs, are moving to opposite ends of the cell, and the spindle fibers in our mitotic spindle are becoming way more pronounced. And so that's kind of a good thing. And then as we actually move, so this would probably be like an early prophase. This would probably be a later prophase. Okay. And then as we move on, all right, the next phase that we actually see in mitosis, which basically, remember, kind of takes about an hour to complete, this is going to be metaphase, where our sister chromatids, okay, that are actually chromosomes now, are going to line up in the middle of that plate. And sometimes, if you're reading or you're watching other things that happen to be there, this is what we actually call the equatorial plate, so kind of like the middle of our cell. But metaphase means middle, and so it always kind of looks like a bunch of chromosomes that are actually lined up, right? Then after metaphase, we move on to anaphase. And anaphase, you can kind of see that the cell is starting to elongate a little bit. But what's going to happen here is that we are going to actually separate our chromosome into sister chromatids. That protein disc, the centromere that holds them together, also allows them to be able to stick onto spindle fibers, is going to start to disintegrate, and that will allow the sister chromatids or the brocros to be able to separate from one another and to be able to start to move to opposite ends of the cell. So the ones that are on the left are going to be pulled by these centrioles on the left until they move over. And then the ones that are on the right are also going to be pulled towards the right hand side of the cell. And so that is pretty much anaphase. I kind of remember it as anaphase away. And then, so here, all right, they're kind of at opposite ends of the cell. And you can see that the mitotic spindle or spindle fibers are much less pronounced as they compare to what they were over here in anaphase. Okay. And but we do have kind of identical numbers and information from both chromatids that are there. Okay, you can also kind of see in the picture that they are showing that, like the nuclear membrane is reforming to protect these chromosomes. You can also kind of see that the chromosomes are starting to be able to unwind a little bit. And then the other thing that's in the back is the nucleolus that's also starting to be able to appear here, as they illustrate telophase here in this video. And then here, once we actually get to it, telophase really is producing those two cells. And then even though they don't show it entirely complete here, let me move myself out of the way here, just so you can kind of actually see it better. Okay. This is actually the process of cytokinesis. And now you can really kind of see how the chromosomes, okay, that are actually in there have really so have really uncoiled and being able to actually look at, they look like little dots that are in there. You can kind of see some of the squigglies that are around here, so that they would be going back to actually kind of looking like they're in chromatin form as opposed to a chromatid or a chromosome form. So they are in there. But some of you were like, wow, they have to be really tiny in order to do this. And I think that this particular like progression of mitosis illustrations that was actually done by Wikimedia, which is, a good site for being able to actually look at some of the things that are there. This one is nice. Does indeed kind of show you just how tiny it is. The nuclear membrane is kind of reformed. This orange dot that's in here is the nucleolus. We're really not going to worry about that. And then you can see that the centrioles have kind of reabsorbed all of their microtubules or spindle fibers are part of the mitotic spindle. Okay, and then cytokinesis happens. So the prefix cyto means cell, kinesis is movement. So we are moving into the cell. So it kind of comes from the top and the bottom, and it is going to push, okay, and until it would actually split into two identical cells. And then these two identical cells would go back to G1 and start the cell cycle all over again. And then these two cells would become four, four would become eight, and so on. Right? So hopefully this kind of helps you look at the M phase of our cell cycle. Okay, so we've now been through G1, we've been through S, we've been through G2, and now we've been through the M phase, and then the cycle would start all over again. I hope this helps clarify things, and I also hope you have a great weekend. I look forward to hopefully being able to be with you guys on Monday. Have a great weekend. Thanks for stopping by.